Yeah, thank you very much. Sorry for the delay. Yeah, it's a real pleasure to be here and share the work that I undertook during my PhD. And yeah, I think we've had a really great introduction already to the world of RNA and why it's important, and it's really preaching to the choir to go into that even more. But we also fundamentally believe that RNA is important in a regulatory sense and in establishing RNA regulatory networks. And amongst that can be sort of a number of mechanisms which can involve the RNA composition itself, as we've heard so nicely this morning, as well as changes in 3D chromatin structure, the RNA binding proteome, interactions with chromatin, and our point of interest, RNA-DNA interaction. And so RNA and DNA tend to share quite a similar space, and we also believe they interact with one another as well. And one of the mechanisms by which this can take place is through the formation of so-called RNA-DNA triple helices. And so these are structures where the double helix of the DNA remains intact, and single-stranded RNA winds its way into the major groove of the helix. And it results in the triple helical structure that you can see on the right-hand side, your left-hand side of the slide. And our hypothesis for how this can lead to the regulation of gene expression is that a regulatory RNA, either in conjunction with an RNA binding protein or independently, can then bind and form one of these structures at a DNA site of regulatory importance, and thereby have a functional consequence, which is usually the form of gene expression. And biophysically, the way this takes place is, and I apologize really in advance to any Dutch members of the audience, is through Hoogstein, or I believe it's pronounced Hoogstein, base pairing. And this allows, theoretically, so you have Watson-Crick base pairing, and then you have Hoogstein base pairing, and this could permit the formation of this triple-stranded structure. And so previous attempts to use these biophysical base pairing rules to predict triplex formation have been made, most notably by the tools triplexator and long target. And they use various different approaches, which I can explain in more detail at my poster later. But they both implement this in their own way. And you think this is a really good idea until you go into the cell. And as we've heard, there's a real dichotomy between what you see biophysics-wise isolated from that cellular context, and once you go back into a cell and look at that data. And so in this benchmarking study from a few years ago, they used MEG3 CHOP sequencing data. And so this theoretically produces the genome-wide binding sites of a single RNA of interest, in this case, a long non-coding RNA MEG3. And then the authors here used either long target or triplexator to try and recall those interactions. And as you can see from the ROC curve on the right-hand side of the slide, there was really room for improvement there, and it shows the dichotomy that does exist between the biophysics and what's really happening in the cell. And so this is the stage that I started working on this kind of topic, and my boss was very keen for us to be able to predict RNA-DNA interactions more accurately. And at about the same time, there was a new method which was published by a group in Heidelberg, which we now term really triplex chip or triplex sequencing. And this involves the genome-wide isolation of either RNA-associated DNA or chromatin-associated RNAs. But you get either a whole genome-wide manner or a whole transcriptome-wide manner. The key word here is or. So it's a real downside of the method. You have to choose at a certain point. So you pull down these structures, and then you choose, do I want to sequence the RNA or the DNA? But what you end up with is genome tracks where you can see which regions of DNA are involved in binding RNAs. And the hypothesis would be that these are involved in binding the regions of RNA, which you can also identify. And it seemed to work. So this panel on the right-hand side of the slide shows the recapitulation of a published triplex interaction between MEG3, the aforementioned link RNA, and a gene locus Col15A1. And the authors of this NatComs paper from a few years ago state that MEG3 binding at this gene locus regulates the downstream gene, TGF-beta receptor 1. And so it looks like triplex sequencing captures these interactions. You just have to be a bit imaginative with it. And so we then asked ourselves, hey, could we use this triplex sequencing data to develop a new sequence-based predictor of triplex formation based on data which is taken from the cellular context, so not purely biophysics. And so we laid out this master plan of how the project would progress and how we would succeed at every step, but it's obviously much easier to put this together at the end of a project after everything's finished, right? But the idea would be that we would identify the sequences responsible for triplex formation from this data by peak calling and then subsequent motif enrichment. And then we would try and learn triplex forming rule sets. So link these kind of unpaired motifs together, see if we could learn rule sets which explain the pairs, and then go on and validate those with genome-wide RNA-DNA interaction data, and also go back to the biophysics and try and validate these structures. And so we went in and we started identifying these sequences, and the process is up on the top left there. And in the end, so at this point, we were keeping sequencing replicates individual because we really weren't sure on how reproducible this data was and how reproducible our approach was either. But what we saw is that for each replicate, we could identify quite a similar number of DNA and RNA motifs, respectively, and that when we used TomTom to look at the similarity of those motifs, they were also actually much more reproducible than we originally thought. So that was nice. We were sort of working our way through step one. We then went on and we collapsed those sets of motifs to remove anything that was identical. We removed, at this point, known protein binding motifs to really try and focus down on things which we thought might purely be involved in RNA-DNA interactions. And then we were left with these sets of motifs, which are down here. So just under 200 DNA motifs and just over 300 RNA motifs. And so then we come to the most interesting bit. So we wanted to figure out if we could take these motifs and pair them up and learn the sequence rules which would make them pair. And to do this, we needed a method from which to learn a probabilistic matrix which would explain or essentially translate an RNA motif into a DNA motif. And what we realized is that we could do that, or we could optimize such a matrix using quadratic programming using a given pair of motifs. We then made an assumption that, okay, if one of these probabilistic matrices is really good, it should explain many base pairings or many motif pairings, sorry. And so when we were doing this and we were taking pairs of motifs, we would then take the mean matrix or the mean probabilistic matrix across all of those motif pairs, and we would think that, that would be what we'd learn. What was really key here, though, was assessing whether these made any sense. And so we needed a really objective way to assess the performance of these probabilistic matrices. And so what we did is we took that motif pair again. So if you have a motif pair and you have one of these translation matrices, which is learned across a set of pairings, we can check how the perfect DNA partner motif would look given that mean matrix as compared to the real sort of assigned partner DNA motif. And if we take the absolute difference between those two motifs, that gives us a really objective value to say, how well does our base pairing matrix describe a given pair of motifs? And then across many pairings of motifs, we can take the mean value of that, and that gives us a way to assess one of these mean code matrices. And so using these principles, we wrote an EM algorithm to try and learn essentially base pairing matrices with a minimized objective value, so that the ones that describe a set of motif pairs the best. And so we did that by initializing a motif pairing, computing our code or our base pairing matrix from that, using our objective value to really assess the performance of that code, and then using the learned code to update the pairing and iterating through this cycle until we reached a base pairing matrix with a minimum code error or objective value. And so as a proof of principle, we simulated some data. So on the top left of the slide, you can see what we did is we simulated 100 pairs of motifs, which we linked with Watson-Crick base pairing. And then we used these and gave them to our EM algorithm. And you can see that across a number of iterations of the algorithm, the objective value on the left-hand y-axis and the number of pairing errors, which is obviously intrinsically linked, both come down with every iteration and eventually minimize and converge at zero. So it was looking good. We had a method which we thought could work. We then took that and we implemented on our triplex motifs. And we were really quite scared at this moment because it's it's quite a large search space. We were working with quite a number of motifs, and we were very scared of the prospect of local minima. And so we knew we really had to fill that search space. And so we ran tens of thousands of initiations of this algorithm to try and fill that search space. And so then we had to try and filter these results and see what really made some sense. And not wanting to lose the link to biophysics at any point in this, we used published RNA-DNA triplet affinities published by a biophysics group in Notre Dame. And we regressed essentially we projected those onto our matrices and we regressed that against our objective value to see if our objective value we were using to assess the performance of our base pairing matrices made biophysical sense. And at the bottom left, you can see we actually got quite a nice regression against those affinities, and it made it look like, hey, our objective value is really describing something which might be biophysically plausible. And when we take the subset of our results shown in the top right of the slide and look at the difference between the biophysical affinity of those results and compared to a size match set of random codes, we were also significantly higher than those. And so then we moved on to the next step. We wanted to really try and implement these in something. And so we took those best performing results and we kind of linearized these matrices and hierarchically clustered them to see what we could see. So could we really find reproducible results amongst those tens of thousands of iterations? And so you can see the results shown in this heat map. And what we did is, after hierarchical clustering and some tree cutting, we ended up with these 8 matrices, all of which came out of those. And although some of these look relatively redundant, if you look into the heat map, they are slightly different. And so that's why they've been come out of different clusters. And so we thought, hey, we've got these basically base pairing matrices. The best way to implement these would be in a local alignment tool, because we can use them as substitution matrices, and we can then use Karl and Hausdorff statistics as a way to statistically say whether we think triplex formation is likely to take place between a user-provided RNA sequence and a user-provided DNA sequence. And so we went on and did that, and putting this together, I realized how chronically short of imagination we were, because we went and called it triplex aligner. And we wanted to go and validate this. And we really needed a new type of data for this. We didn't want to go back to triplex sequencing because we knew it was unpaired, and we weren't quite sure which sequence went with which, or which DNA went with which RNA. And kind of the academic gods answered our call, and during the course of this project, there are a couple of new methods published, namely radical sequencing and RED-C, and these provide one-to-one RNA-DNA interactions. And so what we did is we decomposed these interactions into their RNA and DNA, and then predicted them with either triplex aligner, triplex eta, or long target. We then, when we analyzed the results of this, we could see that triplex aligner quite, outperformed the other tools relatively well, and that was across both human and mouse data, although it really has to be said that these other tools are still useful. They still performed much better on this new data type than on that original trop sequencing data, which I spoke about earlier. And so it's really important to say that. And finally, I know we're a little bit short on time, but we went back to the biophysics. And so we really wanted to kind of close this loop and show, well, essentially not get into any arguments with the biophysicists as well. And so we took our maximally predicted sort of subsequence from that radical sequencing computational validation, had those oligonucleotides synthesized, and then we gave them to our collaboration partners and said, please show us that these form a triplex and we haven't been barking at the wrong tree for the past 3 years. And so I'm really not a biophysics expert, so please, anyone can correct me if I describe anything incorrectly here. But they subjected that to initially a melting analysis on the top right of the slide, and could recover 2 melting points when we provided all 3 of our oligos to that, which implies that first the RNA melts off the triplex and then the DNA double strand melts. In an electrophoretic mobility shift assay, we could see that when we provided these 3 nucleotides, a structure forms which is insensitive to RNase H, which is a really classical signal of triplex formation. And in circular dichroism spectroscopy as well, it produces this form which is unlike either double-stranded DNA or an RNA-DNA hybrid. And so we published that at the end of last year, and we implemented it as an R package, which is available, but in the sort of spirit of the Red Queen hypothesis, where you have to keep running as fast as you can in order just to stay in the same place, we're continuing to develop. And also, we want to really go on and try and use triplex aligner to show that triplex is important. And so now we can use it to look at RNA-DNA bindings and identify putative targets of RNAs in a genome-wide manner, as well as looking at regions of RNA which we predict to be involved in triplex formation. And we've got active collaborations going on to probe those kinds of regions for things like secondary structure. And that's very exciting. I'm looking forward to seeing those results. And we're also continuing the development on the computational side to try and make it into a truly sort of scalable genome-wide tool for the wider scientific community to use. And so I'd just like to thank all of my colleagues at the Institute, as well as the biophysics guys, for all of their hard work on this, as well as the mentoring and supervision of my boss, Ralph Brandes, and also Marcel Schultz, and all of the funding bodies and consortia that enabled this project to take place. And any questions, I'll try and answer.
Time for a question or two.
I can hear you, it's okay.
So you have those matrices that are two sides.
Yeah.
Okay. So I was just wondering, right? So I guess for predicting this, you're assuming independence between those positions.
Yeah.
So did you try to relax that? That's like,
No, so at some point we haven't, but that's really interesting. It's something we're working on implementing, because I was at the RNA meeting in Boulder last year, and Joan Stites gave this unbelievable keynote, and she was talking about RNA triplex, and that you have these kind of mixed models of how they form, and they have sort of sequences in amongst them which really don't form triplex and can reset it. And so we also wondered if that's some of what we're covering, because we're really not sure. We went into it extremely naively, and so at some point we assumed all of these base pairing matrices we're recovering are involved in triplex formation, but it might just be you're recovering other elements of that, so regions around it which prepare that kind of sequence for triplex formation and things like that. So it's something we're actively working on and trying to implement. But thank you.
And so regarding your iterative method, do you know if it is guaranteed to converge to a global optimum, or is it only guaranteed to get you a local?
So it gets you a local, which was a big issue, right? So we had a lot of space, and I eventually plotted it, and you could see all these local minima, and it really scared the living daylights out of us, because we thought we'd never get to that global minima, and that's why we had to initialize it so many times, sort of as many times as feasible. I think I went on holiday one time and just left it running and sort of came back to it. But we hope we found a global minimum. We can't be sure. Yeah. Hopefully, yeah.
Are some RNAs more likely to be involved in these triplexes?
Yeah, so that's something we're now working on. So we're looking. So my poster has kind of what we're working on now, which is looking for the real importance of this, and that involves looking at a lot of RNAs. And you have really classical RNAs like MEG3, which has come up a few times and is a published triplex forming RNA. And if you run it in triplex aligner, it comes up with a mad number of hits. It's huge. It's everywhere. You can take other RNAs and you'll get absolutely nothing. So at least on a computational basis, it looks like there's a difference. However, I imagine it's extremely cell type dependent as well. And because we're predicting it, we're not taking into account an awful lot of subtle things which will change that. But it's something we're working on, and we don't know what makes a triplex forming RNA and what doesn't. It's a real problem. I think wet lab and dry lab will work on it. We also have a lot of collaborations to try and approach that.